I'd like to actually spend some time just introducing our guest speaker. Let me tell you a little bit about Katrina Rogers. I met Katrina I'm going to read her formal bio. She's a leadership expert, and she has a consulting company. She's got over 25 years of experience developing and manufacturing medicines and related technology. She's an expert in laboratory and technical operations. She is an ASQ certified manager of quality and organizational excellence and pharmaceutical GMP professional. Her professional services firm, Katrina Rogers Consulting, specializes in dramatically decreasing the cost and time of bringing new medical products to market. I see that she would do all of those, but the other thing that she is absolutely amazing at is advocating for her community. And so Katrina's in the Spokane region, and she is very involved in the business development component, fostering manpower in the health sciences technology space. She's really big into looking at alternate ways of providing education that may be slightly non-traditional, but help us to get our workforce. We've got thousands of jobs in Washington state that are unfilled because we have unqualified people. And so she's been involved in recruiting, training, thinking about how to do things differently. Most recently, there is a Washington State Summit on how to look at STEM and life sciences in the region, and Katrina was a keynote speaker as well as an organizer of a panel. If you talk about any major organization that's advocating for our scientific life sciences community, Katrina is on it. And able to network to people to make the relationships that are probably most successful in us driving change. But today she's not talking about that. She's talking about her more expertise in clinical laboratory amendments and the relationship that CLIA organization has with the FDA. And I'm going to turn it to Katrina to finish introducing herself and to just highlight the topic for today.
Well, thank you very much, Elizabeth, and I'm really excited to be here today. Let me go ahead and share my slide deck so we can get started on this topic. And you are welcome to, let me see, I need to pop open the chat here so I can make sure that if we have some questions there, they will, if you want to go ahead and put them into the chat. If I spot them while I'm speaking, I will go ahead and answer them then. If I can't, I will be sure to take a look at the chat and check on them at the end. And there will certainly be an opportunity to for you to ask questions at the end. So thanks again, Elizabeth. That's a very kind introduction, and it does seem like I get invited to be in all the best parties, and the ORCA meeting, of course, is one of those. Elizabeth asked me to share with you my thoughts on the impact that the clinical laboratory improvement amendments have on in vitro diagnostic testing. And so that's the topic of our call. Let me see, I've got to get this going. Okay, here we are. And I want to thank the team for inviting me to speak. I really appreciate the opportunity to share my thoughts with you. I've included my contact information on this slide. It will also be on the last slide. But I'd like to also add to Elizabeth's introduction by mentioning that my purpose with my professional services firm is to help people change the world with their medicines and medical devices. And so I do that in a number of different ways, but working on talent development is just, I think, just one of them. So let's go ahead and get started. And I think first, let's set the stage for where we're at. You're all a diverse group of clinical, regulatory, and quality professionals. I believe there's also a few educators and healthcare professionals among you, and you all have an interest and expertise in the regulatory aspects of pharmaceuticals, medical devices, biologicals, and life sciences. And you're all here in the Pacific Northwest. So one of the things I appreciate about this organization is that you have regular meetings to discuss topics of interest and to improve your education. And this, of course, is the October meeting. Let me take a couple of moments here just to thank everyone on the call, and especially anyone who is actively involved in providing healthcare. Really very much appreciate all the effort that you've put forward this past year to help keep us safe, and appreciate the continuing effort to develop new medicines and new tests for this pandemic. So you are aware that because of the pandemic, we've seen more information and more mention of CLIA in the press as it relates to testing for COVID-19. You may also have faced many of these questions in your everyday practice. Your expertise is likely in pharmaceuticals, biologicals, or medical devices, probably not all three, but you may also have, and you may or may not have any expertise with clinical operations where the CLIA regulations really dominate. So some of the questions you may have been faced include, why are there actually two systems? Because there appear to be two systems for regulating in vitro diagnostic tests, and how do laboratory developed tests work, and really how the CLIA regulations interact with the FDA regulations. And really, if you're talking to people who are developing the test, which I do quite a lot, they're always asking me, which of these systems should we actually use? Because there are pros and cons to both. So my purpose here is to share with you, you've come here to get a basic understanding of how the CMS regulations work with the FDA regulations with respect to in vitro diagnostic testing. And I can only give you, in this space of time, kind of an overview on it. As you know, regulatory science is pretty dense and complex, and there's lots of special elements to it. So that's where we can dig into this a little bit more in the questions. And of course, I encourage you to learn more yourself. So in this presentation, the first thing I'll share with you is what the CLIA regulations are. Then we'll talk about how they work with the FDA device regulations to really cover in vitro diagnostic testing. And then we'll talk a little bit about why this really is important now, which goes beyond just understanding how testing works and into some very regulatory related questions that have popped up recently. So let's start out with what are the CLIA regulations. CLIA simply stands for the Clinical Laboratory Improvement Amendments. These amendments were added to the Public Health and Safety Act in 1988, and their purpose was to ensure quality testing for human diagnostic samples, and those samples were the purpose of those samples, of course, is to assist healthcare professionals with making correct diagnoses. So your doctor, of course, when they order a diagnostic and you provide the sample, of course, that goes to the lab, when they receive those results, they want to know that they are accurate and representative of a true diagnostic outcome. More importantly, the laboratory wants to make sure that they get paid, and the payers, including the Centers for Medicare and Medicaid Services, as well as insurance and other payers, want to make sure that when they pay for a test, those results are what they expect. In other words, there's good value there from the test result. So CLIA establishes the quality system for clinical testing laboratories. These amendments are administered by the Division of Clinical Laboratory Improvement and Quality, which is under CMS. And you may or may not know, but CMS really dictates all of the, how things get paid. So the whole point of this is without following the CLIA regulations, laboratories don't get paid. And that's the real pull part of the CLIA regulation. So laboratories have specific requirements under CLIA, and the first one is they must be certified before they can release results, of course, and before they get paid. Those certifications are based on the complexity of the tests that they perform, and the CLIA regulations also establish the test complexity system. Waived tests are simple tests, such as urine dipstick analyses or blood glucose measurements with a simple meter. An example of a moderate test might be a simple immunoassay or a spectrophotometric test. And a high complexity assay, a good example might be a genomic screen or a microarray test. The requirements for laboratories only performing waived tests are fairly simple, and they can receive a CLIA certificate of waiver with a simple application and demonstrating that they are operating under good laboratory practices. Laboratories that perform tests of moderate or high complexity have additional quality system requirements, and they must demonstrate that they've transferred the methods effectively. They must continue to perform them effectively and demonstrate that through performance testing with blind samples. And they also must maintain a quality system. The process of certification starts out relatively simple with an application to your state agency, and then followed by an inspection. And once the inspection has demonstrated that the laboratory is performing correctly, they will receive their certificate. And there's an example of the certificate shown on this slide. Recertification for moderate and high complexity labs occurs every 2 years and is a similar process, though the inspections are less frequent than, say, GMP facility inspections for the FDA. A good point to make here is that Washington State, along with New York State, have their own regulations for laboratories. They follow CLIA generally, but they have some additional changes. So it's important that laboratories in both of those states pay close attention to what their state regulations require. The CLIA regulations also establish the complexity of tests, and they add an additional category of tests called laboratory developed tests, or LDTs. LDTs are tests which are designed, manufactured, and only used in the high complexity lab that created them. Under the CLIA regulations, an LDT is analytically valid, and that's very similar to GLP or GMP analytical validity, where you establish a limit of detection, a linear range, accuracy, and precision criteria for the test. There are no regulations under CLIA for clinical validity. That validity is established by the lab that developed the test. A good example of a laboratory developed test, or LDT, is this microarray test that's shown on this slide. So simply put, what CLIA are regulations that oversee the quality system for clinical laboratories that produce diagnostic test results. They require laboratories to be certified to perform the tests that are on their certificate before they can release results or get paid by Medicare or Medicaid. They also establish the complexity system for tests, which really just talks about how difficult is it for the test to be performed in any particular laboratory, and other quality system requirements. And they allow for high complexity certified labs to develop and release results from their own LDTs. So how do those CLIA and FDA regulations interact? So firstly, these regulations are all established under different legislative mandates. You'll remember that I mentioned that CLIA was established under the amendments to the Public Health Service Act. The FDA regulations are established for medical devices were established in 1976 through an amendment to the Federal Food, Drug, and Cosmetic Act. And so that means that the regulations are under different CFRs as well. So CMS regulates the labs under 42 CFR Part 493, and the FDA regulates the actual tests themselves under 21 CFR Part 809. The 1976 amendments to the Food, Drug, and Cosmetic Act established the 3-tier risk-based system that the FDA uses to manage medical devices. And in vitro diagnostics are, of course, considered a medical device. That it also established the quality controls required for the different categories of medical devices. And I'll take a moment to point out that there's a difference here between the complexity system for the tests and that's used by CMS and the risk-based system that's used by FDA for devices. Complexity refers to how difficult the test is to perform, and the risk elements or the risk categorization is based on the risk to the patient with respect to the safety and efficacy of the actual test itself. So it's possible to have a high complexity test that is moderate risk, or it's possible to have a moderate complexity test that's higher risk. So those systems are parallel, are definitely parallel systems. We can't confuse them both. So all CLIA labs, all CLIA certified labs use FDA cleared and approved tests, right? Depending on the FDA pathway. Labs with high complexity rating or certification may develop their own LDTs, but they can only be used in that same lab where they were developed. They can't be used in other labs because the analytical validation is only for the lab that developed them. And labs are allowed to release results only for the tests that are listed on a certificate. So if a laboratory has created an LDT, they have to modify their certificate in order to add that. Any laboratory that wishes to add some new tests has to modify their certificate in order to add them. The exception being waived tests, where the laboratory with a certificate of waiver is permitted to operate or to release results for any waived test that they have. It's relatively simple to look up the complexity level for a test using the FDA's database. The agency was given the authority to assign those complexities starting in 2003 to help make it a little bit easier. It was a little confusing having to go to CLIA to get your complexity level and then go to the FDA to get your clearance or your approval. So listeners will already have detected that the CLIA regulations allow for an additional marketing pathway for in vitro diagnostic tests. You will likely have spotted that tests of moderate complexity are still required to go through the FDA clearance or approval pathways. Tests of the waived complexity tests, developers could now have the opportunity to submit a dual 510K CLIA waiver because the FDA is now in charge of assigning the complexity of tests. So they can submit one packet of information and get both their 510K clearance as well as their CLIA waiver for the test. For a high complexity test, a developer can choose to use the traditional FDA pathway. But if they are a high complexity laboratory certified under CLIA, they have the option to add that test to their certificate and then market it as a test that they perform themselves. And so that's the pathway that many companies have used in order to introduce their new tests of late. So just to review how the CLIA regulations work with the FDA device regulations. The CLIA regulations regulate the laboratories and their quality systems. The FDA regulations regulate the actual safety and efficacy of the device. All CLIA laboratories use FDA cleared or approved tests, and some of the high complexity certified labs have their own lab developed tests. But any laboratory can only release results for the tests that are on their certificate. Plus, the CLIA regulations allow developers of high complexity tests to market their pathway through their own laboratory where that test was developed. So let's go on to discuss why both systems are important right now. First off, while the FDA has had the authority to regulate laboratory developed tests since 1988, when they were established, they have not generally enforced those controls in the past. And there are three good reasons for this. Firstly, the early LDTs were relatively simple. like this dipstick test that's shown on the slide. They were also available only to a limited population. However, this situation has changed lately, where LDTs have become somewhat more complicated, and they're, with lab centralization, they are available through to more patients. Oh, I see the question from Anjana. Yes, Anjana, DEN is in fact de novo. All right, thank you. Secondly, the FDA often tells us that they are aware that regulatory flexibility is very important for encouraging innovations in this space. And innovations in medical diagnostics are very important to help us detect not just new diseases, but also diseases that have troubled us for a long time. A great example would be the use of high throughput sequencing and next generation sequencing in the diagnosis of cancer and in personalized medicine diagnoses. Thirdly, it takes time for new regulations, the impact of new regulations, to be felt upon existing ones. And of course, we're all very aware that regulatory science is a very slow advancing beast. We have also seen lately a rise in the number of commercial LDTs that are marketed under CLIA. And I've got some examples for you here. The Gallery test is an early detection test for up to 50 kinds of cancer from a single blood sample. This test has been validated in the laboratory and is available to doctors via the CLIA regulations. And the manufacturer is very careful to note that any of these early results need to be verified with additional testing. The test itself detects differences in DNA methylation patterns that are indicative of early forms of cancer. The next example is Progenity's pre-occludia test, which is designed to rule out the condition of preeclampsia in pregnancy, something that affects many pregnant women. This test, while currently in development, does appear to be heading towards the market under the CLIA pathway, at least at first. And it detects the placental growth factor, the amount of placental growth factor, as a biomarker of preeclampsia. Lastly, and the big elephant in the room here, the Theranos Edison, which you may have heard about in the press. This device was designed to detect from a single blood spot hundreds of different kinds of medical conditions all at once. It turns out that we have learned that the Theranos Edison never worked in the way it was intended, and the results that were produced by the Theranos lab were actually produced by more traditional methods and requiring additional samples beyond the spot. But of course, this laboratory developed test is no longer on the market, but it will continue to be a scrutiny because of the very high profile criminal fraud trial that is underway right now. So lastly, I think we can expect practically more regulatory scrutiny. And first off, the FDA has since 2010 been gathering public comment and information, publishing white papers and other discussion papers about laboratory developed tests. And I think you will all agree with me that the comment from the latest paper in 2017 is pretty consistent with the FDA's approach to moving towards an increased regulatory framework. Also, the House of Representatives and Congress have become more interested as well. And we've seen the introduction this year of a bill designed to amend the Food, Drug, and Cosmetic Act to increase the regulation of laboratory developed tests. And of course, the Theranos trial will continue to influence public opinion and public policy. I can say, having looked at some of the evidence from the trial, that it seems to me that we can conclude that there was some regulatory wrongdoing that happened, but that was relatively limited, at least based on what evidence has been presented publicly. Most of the problem there in the Theranos trial appears to be fraud based on poor due diligence, I think, on the part of investors. But I would be very interested in hearing other people's opinions about that topic. So of course, why is it important to understand the CLIA regulations and how they interact with the FDA? Both because the FDA has in the past not strictly enforced their oversight of laboratory developed tests, that that there are an increasing number of laboratory developed tests being offered to the market, and that we can expect to see more regulatory scrutiny, both from the increase in the number of questions as well as the type of information that's coming out from high profile criminal trials. So to conclude, I know you came here with many questions today about what was CLIA and how does it apply. You expected to hear a little bit more about the landscape between the CLIA regulations and the FDA device regulations. And I hope I've been able to show you a little bit about what CLIA is, how it works with the FDA regulations for devices, and why that might be that's a topic of some importance for us today. I would be very interested in your questions as well as your thoughts on this topic. And of course, here's my contact information as well. Okay. Does anyone have any additional questions or comments? Has anyone been following the Theranos trial? I'd love to hear your comments on that.
I definitely have been following the Theranos trial. Thank you for your formal part of your presentation, and we'll make sure that those slides are available. If you want to stop sharing your screen, then we'll see a bigger picture of everybody in terms of answering some questions. So I'm glad that you covered this topic. So there are several different documentaries on the Theranos trial that are going on at the moment. My podcast comes up every week with some new understanding on that. I will only ask one quick question, and this is not related to the majority of your topic about how these regulatory things go together, but your last comment, which is, was there an oversight problem here? And the oversight problem, could it have been a regulatory oversight, or was it an oversight with regards to the investors? And certainly it would be very optimistic in our world of new innovators to actually have someone who was didn't finish Stanford, was 20, I believe, when she started this company, and to achieve such a large initiative that could change the way we really looked at care. Testing, I should say. Could you just comment a little bit about, you mentioned there'll be regulatory changes. What other changes do you think that will come out of this?
I do think that there will be, I think there will be some other regulatory changes, but I suspect that those may be with respect to SEC regulations rather than FDA or CLIA regulations. I think what I've seen in the testimony so far is that it appears the laboratory director that they chose was technically qualified, but not appropriately qualified. So in other words, did not have any, it was definitely a licensed physician and had gone to medical school, but did not have any experience with clinical laboratory operations or releasing results. So there was an element of a gap there. I think that's going to definitely be something that will be of increased scrutiny, I think, on upcoming certifications and recertifications. So that's practically where I think those regulatory changes are going to happen. They will likely happen on the existing labs and labs that want to become a high complexity certified lab going forward. They'll see some increase in questions and some very pointed questions around some of the topics that come out of the trial. I think that from an SEC point of view, it's possible that they may find that the regulations that they have as they exist really should have protected the investors as much as possible, right? That there really wouldn't be much more needed. I think that if you are an accredited investor, you're stepping into some deeper waters anyways, and you should be paying attention closely to what you're looking at. What's interesting are the folks the experts who are testifying. These were people who were evaluating the paperwork and the documentation for example, for Pfizer or for some very experienced investors. And it's interesting, they all chose to pass because they didn't see in the documentation the evidence that the reliable evidence of the quality and efficacy of those tests that they were looking for.
Thank you for that comment. My other area that I'm involved in, as Katrina knows, is angel investing and teaching due diligence on how people might invest in health. And so the comments that you're making with regards to being an informed as well as accredited, anyone can say they're accredited, but to be an informed is something important. So we've got a question in the chat here, which is a pre-submission meeting required for a dual 510K and CLIA application?
No, a pre-submission meeting is not required. A pre-sub itself, right? A pre-sub letter may be appropriate to confirm, but if you have, for the most part, a dual 510K and CLIA application really focuses on those waived tests anyways. So the questions you would have to ask is, would this test be considered waived? And generally, it's fairly obvious from the type of test whether or not it's appropriate for a waiver.
Here's our next question. Oh, sorry. Go ahead.
Sure. I see that Dion was asking about, do the LDTs for COVID-19 under the EUA pathway need to follow CLIA regulations? Yes, they do. Absolutely. Right. So you have to keep in mind that it's the laboratory that developed the test that has to perform the test, and the laboratory itself has to follow CLIA regulations. The test itself, if it has been authorized under the FDA's EUA pathway, then it follows essentially that's the early pathway for an intent to move that test through the FDA's approval or clearance process. And good point, emergency use authorization, that's what EUA stands for. We use these acronyms so readily that I always forget to define them.
I'm sure our audience is familiar. I just put it in there to make sure, because So Linda, okay. You can read Katrina.
Sure. The FDA did not want the Seattle flu study to use their tests to see if SARS-CoV-2 was present in their study samples in March 2020, which doesn't seem wise. Comments. Okay, I actually read a little bit about this, and what it's interesting is the resistance wasn't based on the test per se. The resistance was based on the fact that the samples were pulled for a different purpose. So the IRB had authorized the purpose of those samples for flu testing, and that's what all the patients were told. This is for respiratory flu testing. And so in order to divert those results, or take those samples and then repeat the testing on them for something else, that would have required quite a bit more patient notification and approvals. That was the FDA's position. Now, in the case of a pandemic, I think this is where the Seattle flu study said, no, we think this is a pandemic situation. It's a public health situation. We're going to go ahead and proceed to get the information. And they did, and it turned out to be highly valuable. What's very interesting about this is the recent release of new guidance, or at least new draft guidance, around using samples that have been collected for different purposes. So I think that I definitely think there's a regulatory shift happening here too, very quietly, around this very specific topic. So Linda, that's a great question.
Yeah, I can understand that. I mean, we put into our informed consents in our clinical trials that samples may be stored for long term for other purposes, and people can either as part of that consent, either consent to it or say, no, I don't want my sample stored after the study's done. So yeah, so that helps me understand it a little better. But so do you have a, is it easy to find that guidance? Because I'd be interested in reading it.
Let me take a look for that. I think it's relatively new. Use of other samples. I think I've seen it posted in the last couple of weeks. Let me make a note on it and I'll drop into my FDA folder and see if I can find it. Is this last week's?
So the looks like
This
there
would
was
be,
a recent
yeah, studies
article,
using
right?
leftover de-identified human specimens require IRB review. So I'll put that, I'll drop that into the chat.
Great, thanks.
You are welcome. I believe there's some other information about that out there as well, but this is the latest item, so I'm guessing it's probably going to reference some of the earlier clinical guidance.
Thanks.
You're welcome. You're welcome. Are there any additional questions, thoughts?
So when you mentioned with the Theranos situation, which I only have a little bit of knowledge about, but about maybe how the investors should have been looking into it a little more. And I think, I mean, I don't know, I mean, Elizabeth might agree or disagree, but since she knows more about the investor part, but it's like, I don't often think of investors of doing that sort of thing. It's, I mean, I guess they always say you should be informed, but anyway, it just seems like it would be maybe people would want to believe that everything was wonderful and put their money into it. And
well, and I think that that's the risk for almost anything that we do, right? So we recognize, and this is one of the reasons why we have ethics boards and IRBs, that people who have medical conditions, serious medical conditions for which there are few to no effective treatments, reach a state where they can be very desperate and are willing to try some things that maybe are clinically inadvisable. And that creates a space where they can be taken advantage of. I think that's really the basis, the whole basis for the Food, Drug, and Cosmetic Act. Almost every regulation we have is situations where people they either get a little over optimistic or they get a little desperate and they're willing to try something out because they want it to be true. And that's, you know, should we protect? I think that's our whole point here, is that we're attempting to protect people a little bit from themselves and from human nature. I will say that with respect to investments in particular, this is why some of the best advice is to really invest in things that you know pretty well, because then you will be more likely to understand not only the prospectus and the information that's presented to you, but you'll be a little skeptical of some of the claims and maybe dig a little bit deeper. If, you know, investors who are not experts in that particular system, and I think, Elizabeth, this gets back to the health angel, you know, wanting to be investing in health technologies, right? The best way to go is to work with a team of people who do have some expertise in that space and who can help you recognize, because we all have a BS meter, and you just have to calibrate yours a little bit more to figure out, to detect the BS that's present, say, in medical devices, as opposed to detecting the BS that is present in an advertisement for an over-the-counter pain relief cream.
So I put a couple of, in due diligence, we look at, and if you just took any lab test or you'd be looking at the team, you know, was this a credible team? Enthusiasm is one, but did they have the right advisors in the clinical space to be able to make a decision, as opposed to, I mean, I think Henry Kissinger was, you know, had some expertise from political persona, but maybe not in this particular area of space. So was the team appropriate? Did they have some market traction? You know, would this fit in with all of the other laboratory offerings? And where did those other laboratory offerings sit in the regulatory space? And what was the pathway, you know, to make sure that some of this was given some consideration? Was the product and prototype working as it was said? And, you know, that's the side-by-side comparison with other standards of care practice in terms of evaluating that. And then what were their financial predictions? I mean, they in terms of optimism. And so that was, anyway, not to turn this into a due diligence topic on this, but in this particular situation, those are the kinds of things you would be looking at. And all of us with our wearing our clinical associate regulatory quality hats would look at specific components of that to drill down to see whether or not they had that in place. So
right. And I would say that the general youth of the team, to my perspective, isn't necessarily a barrier, right? But I think a big piece of it is, you know, what is the consistency of the message? What is being said? As Elizabeth points out, right? You're looking at the whole picture, and these things they're not easy to understand, right? I'm a lab expert in laboratory operations, and I've done a lot of work in analytical chemistry, and I can tell you that it's very easy to put together a report that looks on the surface pretty smart, pretty sharp, you know, like this. Here's a valid assay. Here's, you know, it's measuring these things. It's very effective from an analytical validity point of view. But when you dig into it a little bit and you look at what they're claiming that the tests are proving in particular data sets, it doesn't really show that at all. So, but it does take quite a bit of digging in order to figure that out.
That's another topic that I wonder if people are interested in is the FDA regulates claims. So there's a section of, you know, your marketing submission and what it is that you can actually say that you're doing. I don't know that we've actually had a talk on what can be included in regulatory claims. And one of the things that we always look at is what does our marketing literature say versus our labeling. And so you might come in on your labeling and say it has all of the exclusions. My kids were forever saying, why can a pharmaceutical company market directly, and did a quick little blurb, and don't use it if you've got this and conditions? And they're going, Mom, who can even listen to that little blurb because they talk through it so quickly, or it's in such fine print at the bottom of an advertisement. So there's labeling claims, and then there's marketing claims, and sometimes they are a little bit in contrast, and you'd want to make sure that they correspond. And so I don't know if anyone else is interested in that. If you're, just raise your hand if you think that would be a good topic for. So
I think it would be interesting. I mean, it's one step beyond where I work. I work in like the phase 3, you know, area, and what happens afterwards is kind of a mystery to me. So that would be interesting.
Yeah. Well, that's a significant criticism too of the industry as a whole, right? That so much more money is spent on this marketing element and really aligning those things and getting the payers on board and using patients to get the payers on board.
That well, I think that's you match that up to, you know, what happened with with opioids, right? You know, and all of the marketing that happened with them, or with any new product. I know, you know, working in HIV for pre-exposure prophylaxis, you know, Gilead had one product that was approved, and now they have a slightly better one than Truvada and Descovy, it's called. And so it gets, now that gets pushed, but it's not necessary for everybody, and they could be taking the generic version of Truvada. So yeah, it happens in a lot of different spaces in the pharmaceutical industry, that there's you know, with the marketing, there's a lot of marketing of the new the new.
So very true. Are
Other
there
questions that
Elizabeth.
other questions that people would like to ask that might relate to what you're currently working on, or are you facing, how many people are working with with CLIA regulatory?
Well, I know that our U.S. labs are CLIA certified, and that NIH considers that to be sufficient in a lot of cases, and that those labs don't need a lot more oversight. So it was interesting. I mean, this was very applicable to, you know, we work closely with a network lab center, and so understanding some of this stuff has been good.
Right. Well, and because a CLIA lab is automatically, they're going to have the quality systems and the operational structure to be a GLP laboratory. So there is the option for laboratories to kind of dip into both of those spaces if they want to. And certainly there was quite a bit of, and Elizabeth's got a great question too. I want to follow up on that. There's certainly a lot of interest in becoming a CLIA certified lab, and very quickly with respect to the COVID-19 testing, right? So we saw a rise in people manufacturing the transport media, right? So many companies got into manufacturing transport media, viral transport media, so the samples could be transported to laboratories. But there was such a dearth of laboratories that even veterinary clinical laboratories were looking to get CLIA certified in order to be able to perform these tests. And it made sense because, again, the requirements for veterinary laboratories are similar enough, and they had to already have a quality system, that it was relatively straightforward for them to get into that part of the market. There was just no desire to do it before the pandemic because there wasn't the demand for the testing. So Elizabeth asked what other countries are doing with regards to regulating lab testings. And I guess what we traditionally call the first world countries, like Canada and the EU and the MHRA, they all have their own systems for regulating quality, laboratory quality testing. Typically, the devices are regulated under a separate part of that as well. So they kind of similarly have sort of a dual system. But I don't think it's quite the tension that it exists here in the United States with respect to the laboratory developed tests.
Thank you for answering that. I'm just curious, as we look at, real world data requirements and new changes about whether or not, if I was a lab test developer, would I find it easier to bring my test to market in the United States versus going to another country?
That's a really interesting question. Mm-hmm. And you were going to say I interrupted you before you finished your question.
Well, I just, in terms of bringing medical devices to the market, it was perceived that it would be easier to go to Europe, and now we know that that's not the case. So I wondered if the same thing happened with the laboratory testing.
So I think that that's really true, right? It looks easier. It's not easier, right? It's similarly, you could say, oh, well, it looks easier to do this in Canada. It's not really easier. It's just different. And it takes a different structure and a different, like for any device in the EU, you have to find a local provider to interact with the regulatory agencies. They have to have their own QP. That there is a structure that you need to follow, and those regulations are also as tricky to navigate as the FDA's are. So I would say that in some respects, if you're a US developer, it almost makes sense to start in the wherever you're at. Start with the regulatory agencies that you have there, because you have to put together a package, even if you are going to go global. And much of that data will still be very useful globally. And Elizabeth, you are also true. Notifying bodies are overworked, partly because the EU has changed their regulations for notifying bodies. So many of the notifying bodies have to get themselves re-accredited and not everyone is going down that pathway. So that there's a turnover in those organizations, and I think that that is creating a bit of a backlog. Brexit, I think, also is creating a bit of a backlog too, because the regulations are staying the same. They're sort of parallel now, and that's going to mean some diversity there. People are still trying to figure that out as well.
I'm typing this in, but I could be just saying it. Our last talk on the FDA regulatory by Michelle Lott talked about whether or not it made sense from a financial perspective to take your product to market. So if you're not anticipating that this is going to be a high revenue, the costs in terms of time, but also financial fees is significant, and it might be worth, if anyone was interested in that dialogue, just going back to refresh that video that talked about what kind of factors would you use to consider whether or not approaching a European organization would be the best thing to do.
Right. That makes a great point, because I think we often forget that there's a business part to the decision as well as the regulatory part to the decision. So you have to stop and think about what's the business risk as well as what's the regulatory risk whenever you're looking at some of these topics. I heard a great presentation within the last month about a very similar thing, where people were talking, the experts were discussing whether or not you should take a pharmaceutical yourself into the EU, right? Whether you should try to launch it yourself or whether you should find a marketing partner. And it was much along those same lines, right? It really depended on overall the size of the market, how much you wanted to spend, and what your overall risk level would be in terms of expertise for that market.
So if we're really practical about the QMS system, is there some suggestion that you might make? Has anyone had someone who was on the call was actually only on for 30 minutes before they had to go off to an audit. So I asked this question about the FDA warning letters that have come out recently, or any audits that have actually occurred with labs recently. Are there any tips that would come out that I, as a regulatory professional, might need to know, or as a clinical associate might need to know? So,
okay. So I definitely, if you knew you had an audit upcoming, you would be wise to evaluate recent audits for similar organizations and kind of see where those questions were. Look at the agencies. They publish their histories or their, the summaries of what observations have been made generally. There are subscription services that are available to help you ferret some of that information out. One of the places I've found really very useful would be the FDA's MAPP, their Manual of Accepted Policies and Procedures. By digging into that for specific topics, you can often find some very interesting guidance with respect to what the inspectors will be looking for.
So I'm just going to put that reference into here.
Absolutely.
So that people actually, if you're not familiar with it, have a handy link.
Right. And I also believe that it's my understanding that the CLIA inspections are typically done by state boards. And so that's, as opposed to an FDA facility inspection, which is done generally by the center that's in your region. So it's a little, there's just a little bit of a difference in perspective there.
So with regards to, I want to be sensitive that if there's any other questions that we get them addressed. If there's any other topics that you'd like to ask Katrina about as she's working in this quality area. Is the QMS system that is required for IVDs similar to that of other medical devices? And are there major differences?
There are not major differences. So the FDA they've historically had their own quality management system regulation, which applied to medical devices, both in vitro diagnostics as well as more traditional devices like physical devices. They have moved in the direction of ISO 13485 certification as being the quality system requirement for medical devices, and that's to encourage, I think, harmonization internationally, especially with the EU. So there aren't really any major differences with respect to with respect to in vitro diagnostics as opposed to other types of physical medical devices. And that 13485 certification is definitely a standard in the industry.
Well, I think that we are going to draw ourselves to a close. Thanks, Dion, for joining us from Canada, from Toronto. I'm happy and hoping that your international regulatory program is as fascinating as it sounds.
Thanks, Liz.
So thank you to everybody for coming today.